Every single person on this planet began as a single cell. One cell, barely visible without magnification, containing the complete genetic blueprint of a human being. And from that single cell, in approximately 270 days, a full human body was assembled, with a brain capable of thought, a heart capable of feeling, hands capable of touch, and a voice that would announce its arrival to the world in a cry that its parents would remember for the rest of their lives. The development of a baby in the womb is one of the most extraordinary processes in the natural world. It is a story that has been unfolding in the same essential form for hundreds of thousands of years of human history, in every culture, on every continent, in every language. And despite all the science that has been devoted to understanding it, despite the ultrasound machines and the genetics laboratories and the molecular biology that has mapped every step of the process, it retains a quality that can only be called miraculous. A quality that comes from contemplating the sheer improbability of what is achieved and the precision with which it is achieved in the SPA, ACE of 9 months. Today, from the perspective of embryology, developmental biology, and the clinical science of fetal medicine, we are going to walk through the entire journey, from the moment of fertilization to the moment of birth, in the specific, evidence-based detail that reveals not just what happens but why it happens, and how the extraordinary architecture of a human being is assembled from a single microscopic beginning. This video is for everyone who has ever been pregnant, known someone who has, is planning to become pregnant, works in healthcare, or has simply looked at a child or an infant and wondered with genuine curiosity how this remarkable thing came to be. Because the science of how a baby grows is one of the most beautiful stories in biology, and most people have never heard it told in full. Let us begin at conception, the moment that starts everything. Fertilization occurs when a single sperm cell successfully penetrates and fuses with a mature egg cell, the oocyte, in the fallopian tube. Of the hundreds of millions of sperm released, only a few hundred reach the egg, and only one achieves fertilization. The moment of fusion triggers an immediate electrical and chemical response in the egg that prevents any other sperm from entering, the cortical reaction, and the genetic material from both sperm and egg combines to form the zygote, the very first cell of a new human being. This first cell is extraordinary in what it contains. 23 chromosomes from the egg and 23 from the sperm combine to create a full set of 46 chromosomes, the complete genetic instructions for every protein, every enzyme, every structural molecule the new body will ever produce. The sex of the baby is determined at this moment by whether the sperm carried an X chromosome or a Y chromosome, the eye color, the blood type, the predispositions of the immune system, the architectural tendencies of the brain, all encoded in this single microscopic cell. Within hours of fertilization, the zygote begins to divide, not growing larger, but dividing the existing cell mass into smaller and smaller units, each carrying the same genetic information. 2 cells become 4, 4 become 8, 8 becomes 16. This dividing cluster, called the morula, travels slowly down the fallopian tube toward the uterus over approximately 3 to 4 days. The journey is not passive. The fallopian tube's inner lining, covered with microscopic hair-like cilia, beats in coordinated waves that help carry the developing embryo toward its destination. By the time the cluster reaches the uterus, around days 6 and 10 after fertilization, it has transformed into the blastocyst, a hollow ball of approximately 100 cells organized into 2 distinct groups. The inner cell mass, a small cluster of cells on one side of the hollow sphere, will become the baby. The outer layer, the trophoblast, will become the placenta and the surrounding membranes that protect and sustain the developing life. This is one of the first and most consequential acts of biological decision-making in development. Cells that are genetically identical choosing different developmental fates, committing to becoming either the person or the life support system that will sustain that person. The mechanisms that drive this commitment involve master regulatory genes called transcription factors that activate different developmental programs in different cell positions within the blastocyst. Some of the deepest questions in developmental biology. Implantation occurs as the blastocyst reaches the uterus and burrows into the endometrium, the hormonally prepared inner lining of the uterine wall. The trophoblast cells extend finger-like projections into the endometrial tissue, beginning the process of establishing the placenta. The inner cell mass begins organizing itself into the embryonic disc, a flat, 2-layered structure that is the architectural foundation of the entire body. The embryonic period, weeks 3 through 8 of gestation, is the most critical and the most astonishing phase of prenatal development. It is during these 6 weeks that all of the body's major organ systems are established in their basic form, arising from a flat disk of cells through a series of folding, migration, and differentiation events of breathtaking precision. At 3 weeks of gestation, the embryo is approximately 1.5 millimeters in length, yet development is already proceeding at extraordinary speed. The process called gastrulation reorganizes the 2-layered embryonic disc into a 3-layered structure: the ectoderm, the mesoderm, and the endoderm, the 3 primary germ layers from which every tissue and organ in the body will be derived. The ectoderm will give rise to the nervous system, the skin, the sensory organs, and the teeth. The mesoderm will give rise to the muscles, the bones, the kidneys, the heart, and the vascular system. The endoderm will give rise to the lungs, the gastrointestinal tract, the liver, and the pancreas. Simultaneously, at 3 weeks, the neural plate forms, a thickening of the ectoderm along the midline of the embryo, and begins to fold, its edges curling upward and fusing along the midline to create the neural tube. This neural tube is the origin of the entire central nervous system, the brain, and the spinal cord. The folding of the neural plate and the fusion of the neural tube is one of the most precisely choreographed events in embryonic development, and its failure produces neural tube defects including spina bifida and anencephaly. Adequate folic acid before and during early pregnancy dramatically reduces the risk of neural tube defects, which is why folic acid supplementation is the single most universally recommended nutritional intervention in prenatal care. At approximately 3 to 4 weeks of gestation, before most women even know they are pregnant, the heart begins beating. It does not begin as the 4-chambered organ that the adult heart is. It begins as a simple tube of cardiac muscle cells that loops, folds, and septates into the recognizable 4-chamber architecture over the following 2 to 3 weeks. But it beats. The rhythm that will continue without pause for the entire length of the person's life starts here, in a structure barely the size of a poppy seed, in the 4th week after fertilization. By week 5, the embryo has grown to approximately 5 to 6 millimeters, still smaller than a grain of rice, yet limb buds have appeared, the precursors to the arms and legs. The optic vesicles, the beginnings of the eyes, are forming as outgrowths of the developing forebrain. The pharyngeal arches are giving rise to the structures of the face, the jaw, the ear, and the neck. The liver and the pancreas are beginning to form as outgrowths of the primitive gut. Week 6 brings one of the most visible and emotionally significant milestones of early pregnancy ultrasound, the first visible heartbeat. At 6 weeks, the cardiac structures are developed enough that a beating heart can be seen on transvaginal ultrasound as a rhythmic flickering at the center of the embryo, at a rate of approximately 100 to 110 beats per minute. For many parents, this is the moment that transforms the pregnancy from an abstract biochemical fact into the living reality of a developing person. The limb buds elongate during weeks 6 through 8 into recognizable arm and leg structures. The hands form as paddle-like plates from which the fingers emerge by week 7 through a process of programmed cell death, apoptosis, in the tissue between the developing digits. Apoptosis, the deliberate, genetically programmed death of specific cells, is as important to normal development as cell growth and division. The interdigital tissue of the hands and feet is deliberately eliminated to reveal the 5-fingered structure beneath. By the end of week 8, the end of the embryonic period, the embryo has grown to approximately 25 to 30 millimeters and weighs approximately 1 gram. All of the major organ systems are present in recognizable form. The face has its human proportions. The limbs are jointed. The brain has its basic 3-part division: forebrain, midbrain, and hindbrain. The embryo is recognizably human. From this point, the scientific designation changes. The term embryo gives way to fetus, reflecting the completion of the period of primary organ formation and the beginning of the period of growth, maturation, and refinement. The fetal period, weeks 9 through 40, is in some ways less dramatic than the embryonic period in terms of the novelty of what is formed, but no less extraordinary in the quantity and quality of development that occurs. In the first trimester fetus, weeks 9 through 13, organ systems established in rudimentary form begin to function. The kidneys begin producing urine, which the fetus swallows, contributing to the amniotic fluid that surrounds it. The liver begins producing red blood cells, a function that was previously performed by the yolk sac. The intestines, which initially developed outside the body cavity due to the rapid growth of the liver, return to the abdominal cavity by week 12 in a precisely timed retraction and rotation that positions them correctly for adult function. By the end of the first trimester in week 12, the fetus is approximately 6 to 7 centimeters in length and weighs approximately 14 grams. The face has its distinctly individual features. The sex of the baby becomes visually apparent in the external anatomy at this stage as the genital structures differentiate under the influence of gonadal hormones. The bones of the skeleton are beginning to ossify, to replace the cartilaginous template of the early skeleton with the calcium mineralized bone of the mature skeleton. The second trimester, weeks 14 through 26, is the period of the most rapid proportional growth, during which the fetus transitions from an entirely dependent recipient of maternal support to an organism with increasingly independent physiological capabilities. At approximately 14 weeks, the fetus begins to make breathing movements, inhaling and exhaling amniotic fluid in rhythmic cycles that exercise the respiratory muscles and the respiratory control centers of the brain, training the systems that will be needed for air breathing at birth. These practice breathing movements are not oxygen exchange. The placenta continues to perform that function, but they are essential preparation for the transition that birth will require. The skin develops its specialized structures during the second trimester: the hair follicles, the sweat glands, the sebaceous glands. The fetus develops a covering of fine, soft hair called lanugo that covers the body from approximately 16 weeks, helping to anchor the vernix caseosa, the waxy protective coating that forms on the fetal skin from the secretions of the sebaceous glands, to the skin surface. Vernix serves as a waterproofing agent that protects the fetal skin from the amniotic fluid during the long months of immersion, and it also has antimicrobial properties that contribute to the newborn's early immune protection. At approximately 18 weeks of gestation, the fetus's auditory system is sufficiently developed to begin detecting sounds from the outside world. The low-frequency sounds of the maternal voice, transmitted through the abdominal wall and the amniotic fluid, are detectable to the developing auditory system, and research has demonstrated that newborns preferentially respond to their mother's voice compared to other female voices, evidence that auditory learning and memory are already occurring before birth. The maternal heartbeat, a rhythmic, constant accompaniment to the entire intrauterine experience, is the background music of fetal life, which is why the sound of a heartbeat is one of the most universally soothing stimuli for newborns. Quickening, the pregnant person's first perception of fetal movement, typically occurs between 16 and 25 weeks for first pregnancies, and somewhat earlier for subsequent pregnancies. Research into fetal movement patterns has documented that fetuses exhibit cyclical activity and rest states that parallel sleep-wake cycling, and that changes in normal fetal movement patterns are among the most reliable clinical indicators of fetal well-being. The development of the fetal senses during the second and third trimesters reveals an aspect of prenatal life that most people find genuinely surprising: the degree to which the fetus is not simply growing and maturing in isolation but is actively experiencing and learning from its environment. Vision is the last of the major senses to develop, and it remains limited throughout fetal life by the absence of light in the uterine environment. The retinal photoreceptors, the rods and cones that detect light, are present and structurally mature by approximately 26 to 28 weeks, but the visual cortex is still developing. However, fetuses do respond to bright external light applied to the maternal abdomen with documented changes in fetal heart rate and movement patterns, demonstrating that the visual system is functional even in the intrauterine environment. The sense of touch develops earlier than vision. Tactile reflexes are present as early as 7 to 8 weeks of gestation. By 24 to 26 weeks, the fetus responds to touch across much of its body surface and actively explores its environment, touching its face, grasping the umbilical cord, making sucking and swallowing movements that are the earliest forms of sensorimotor learning. The sense of taste is operational prenatally. The fetus swallows amniotic fluid throughout gestation, and the taste compounds dissolved in that fluid, influenced by the maternal diet, are detected by taste receptors that are functional by approximately 14 weeks of gestation. Research has documented that fetuses swallow more amniotic fluid when it is sweetened by maternal ingestion of sweet-tasting foods. This prenatal exposure to flavor compounds has been proposed as one mechanism by which food preferences established in the intrauterine environment may persist into postnatal life. Flavor learning before birth. The concept of fetal programming, the hypothesis that the intrauterine environment during critical periods of development shapes long-term health outcomes through epigenetic and structural mechanisms, has become one of the most clinically significant areas of perinatal research. Pioneered by epidemiologist David Barker, whose work on the developmental origins of health and disease revolutionized thinking about adult chronic illness. The field has established that nutritional, hormonal, and stress-related exposures during fetal development can permanently alter gene expression patterns, organ development trajectories, and metabolic set points in ways that influence the risk of cardiovascular disease, diabetes, hypertension, and metabolic syndrome across the entire subsequent life course. Before we continue to the final weeks of development, here is a pause for you. Whether you have been through a pregnancy or are simply learning about this process for the first time, what part of this development story has most surprised or moved you? Drop a comment below right now. This is one of the topics where the community's response means the most to us, because the story of how a human being begins touches something universal. We read every comment. The period of viability, the gestational age at which a baby can potentially survive outside the uterus with intensive medical support, is currently considered to begin at approximately 22 to 24 weeks of gestation in centers with the most advanced neonatal facilities. The threshold of 28 weeks represents a point at which survival rates become substantially more favorable, and babies born at 32 weeks or beyond have outcomes that approach those of term infants with appropriate neonatal care. The third trimester, weeks 27 through 40, is the period of the most dramatic absolute weight gain and of the critical maturation of the systems required for independent survival at birth. Above all, the lungs. The lungs undergo a complex sequence of branching to create the extraordinary tree-like architecture of the approximately 300 million alveoli, air sacs, of the mature lung. The critical limiting factor in lung maturation is the production of surfactant, the mixture of phospholipids and proteins produced by specialized cells called type II pneumocytes that lines the alveolar surface and reduces the surface tension that would otherwise cause the alveoli to collapse with each exhalation. Surfactant production begins in meaningful quantities at approximately 24 to 28 weeks of gestation, reaching levels adequate for sustained air breathing in most fetuses by approximately 34 to 36 weeks. The administration of corticosteroids, betamethasone or dexamethasone, to women at risk of preterm delivery between 24 and 34 weeks of gestation accelerates surfactant production and is one of the most evidence-supported interventions in all of neonatal medicine, reducing neonatal respiratory distress syndrome mortality by approximately 40%. The brain undergoes its most dramatic architectural transformation in the third trimester, the development of the cerebral cortex's characteristic folded structure. Over the final trimester, the rapid proliferation of neurons in the upper cortical layers and the mechanical constraints of the skull produce the dramatic gyrification, the folding that creates the ridges and valleys visible on the surface of the mature brain, and that dramatically increases the cortical surface area accommodated within the skull's volume. The immune system receives a critical boost during the third trimester through the transfer of maternal antibodies, immunoglobulin G, across the placenta in a process of active transport that accelerates in the final weeks of gestation. These maternal antibodies provide the newborn with passive immunity against pathogens that the mother has previously encountered, protecting the newborn during the months before the maturation of its own active immune response. Fat deposition accelerates dramatically in the final weeks of gestation, from approximately 2 to 3% of body weight at 28 weeks to approximately 15% at term, providing the energy reserves and thermal insulation that the newborn will need in the first days of independent life. By week 40, the estimated due date, the average baby weighs approximately 3 to 3.5 kilograms, measures approximately 50 centimeters in length, and is physiologically prepared for the extraordinary transition that birth demands. The lungs are mature, the liver is producing bile, the kidneys are producing urine, the adrenal glands are producing cortisol. The thyroid gland is producing thyroid hormone. Every major endocrine axis is functional. The nervous system is capable of coordinating the complex reflexes, the rooting reflex, the sucking reflex, the Moro reflex, that will be the newborn's first behavioral repertoire. And then labor begins. And in a period of hours, the baby that has spent 9 months in the protected, warm, fluid-filled world of the uterus is born into the vastly different environment of the external world. The first breath, the first cry, the first contact with the people who have been waiting for it. The newborn, taken in arms for the first time, is the culmination of a biological process of extraordinary complexity and extraordinary precision, a process that has assembled from a single microscopic cell, a complete human being with the full complement of organs, systems, and biological capabilities needed to begin a life. The first hours of life outside the womb represent a biological transition that is as remarkable as any single moment of the 9 months that preceded it. In the span of minutes, the newborn must accomplish what took the fetus an entire gestation to prepare for: the shift from placental dependence to independent physiological functioning. The first breath is the initiating event. The compression of the chest during passage through the birth canal expels some of the fluid that has filled the fetal lungs throughout gestation. With the chest's recoil after delivery, the first negative intrathoracic pressure is generated, drawing air into the lungs for the first time. The surfactant coating the alveolar surfaces, produced and accumulated throughout the third trimester, maintains the alveoli open against the surface tension that would otherwise collapse them with each exhalation. Without that surfactant, the first breath would require enormous effort, and the lungs would progressively fail. The pathophysiology of respiratory distress syndrome in the premature infant who lacks adequate surfactant. With the first breath comes the most rapid cardiovascular reorganization in human biology. Throughout fetal life, the lungs were non-functional as a gas exchange organ, and the fetal circulation bypassed them through 2 shunts: the foramen oval between the heart's upper chambers and the ductus arteriosus connecting the pulmonary artery to the aorta. With the first breath, pulmonary vascular resistance drops precipitously as the lung vessels dilate in response to the oxygen now flooding the alveoli. Blood rushes into the newly aerated lungs. The pressure relationships in the heart chambers reverse. The foramen oval closes functionally, sealed by the pressure change, and the ductus arteriosus constricts in response to the rising oxygen tension of the blood. Within hours, the circulatory organization of the rest of a human life is established. The newborn that emerges from this extraordinary process of transformation carries within its body the record of 9 months of biological achievement. Every cell, every organ, every synapse, a testament to the precision and robustness of developmental biology at its most extraordinary. The baby in someone's arms for the first time is not simply new. It is the product of one of the most complex construction projects in the known universe, executed without blueprints, without supervisors, and without error tolerances that would be acceptable in any human engineering endeavor. From 1 cell, 270 days, 1 person. The miracle is not a metaphor. It is the most literal description of what biological development actually is. 270 days, trillions of cell divisions, hundreds of billions of neurons, dozens of organs, 1 person. The science of how this is accomplished is one of the most beautiful stories in all of biology. And now you know it. If this video gave you a deeper understanding of the miracle of human development, from the first cell to the first breath, please support this channel. Hit the like button right now, subscribe so you never miss a video, and leave a comment below. Tell us what this video made you think about, whether it changed how you think about pregnancy, or whether it brought back memories of your own experience welcoming a new life. We read every comment, and this is one of the conversations we are most grateful to be part of. Thank you for watching. We will see you in the next one.